Hello guys, welcome all. Okay, this is breaking news. I am sharing my screen. This is the third video of Moonlight Biology. And the breaking news is about the release of the new Alphafold 3. For the people that does not know about Alphafold, Alphafold is a AI-based protein predictor, and we were previously in AlphaFold version number 2, but right now there has been submitted in Nature accelerated preprint, I think it's called. Well, I can share you. It's called accelerated article preview, but actually there. It's very cool news because now we will be able to predict complexes of proteins, nucleic acids, and small molecules with the addition even with of ions and modified residues. So this is super cool news. I will try to go as fast as possible so you don't get bored. But this is actually the preprint, as I am telling you. Sorry, not the preprint. Is the how is it called? Is the accelerated article preview. Actually, you can see here in this line how it says accelerated article. So this means that it's like it has to be published as soon as possible. But the interesting thing is that it was just published right now, May 8, 2024. The first authors are Josh Abramson et al. And John M. Jumper is the last author. But this is very interesting. As you can remember, AlphaFold 3 is based. Well, the creators are the Google DeepMind from London, UK. And let's see very quickly what about the figures. But it seems that obviously, because I have not seen the article yet, but as you can see here, it seems that we will be able to generate like complexes based on DNA or DNA and protein, or even it seems that you can generate RNA and protein, or even the tree of the complexes based on the three biomolecules. So this is like all the things that they did. I will try to see them very quickly. But usually this is very cool. You can see here on figure number in figure A. This is figure A from, I don't know, it should be figure A from probably figure number 3. But you can see here that this is a tRNA, and the purple probably will be the RNA generated with the AlphaFold 3 AI. And this is in complex with another protein that it's in gray. This is the structure, usually that it's the one that it has been already tested via experiments. And the blue line probably will be the predicted based on the AI. So let's go very quickly to the other part that it's the AlphaFold server web page that I have just logged in. So it would be very easy because you just have to log in using your Google account, and then you just have to sign very quickly into this AlphaFold server. But let's see very quickly what it can do. So I will not put right now the video of Google DeepMind, the AlphaFold version 3, but I will go directly to some examples like this one. You can see this is an example where they put it into the web page of the AlphaFold server beta, and you can see here that they generated a protein, RNA, and ion complex. They generated also a protein glycan ion and a protein DNA ion molecule complex. So this is very cool. There's also frequently asked questions, like for example, what biological molecule types can be modeled. So as you can see, it could be protein, DNA, and RNA, but you can also add ions like calcium, magnesium, and so on, like zinc. And you can also add modifications on the proteins like phosphorylation, acetylation, palmotylation, and so on. So it's super cool news. That's why it's like a breaking news in the molecular biology field. Also, you can see that you can do the same with the DNA and RNA. You can add methylations, formulations. On RNA, you can do all these kind of things. There's also a limit based on the job that you input every time. So it seems that a protein is 1 token, DNA also 1 token, and so on. You can run 10 jobs per day. and you can I will go very fast through the facts, but it says, like, for example, inputs, it's a protein single letter, or you can upload a FASTA file. That's very cool. You can enter DNA single letter nucleotide, and RNA. You can also enter the single nucleotide sequence, and you can add the ligands based on the chemical code. So is there any restrictions? It seems that there's like restriction based mainly in viral pathogens. So probably you will not be able like to upload and generate molecules based on like virus, like like COVID or so. So this is important, but it seems that there's no like it seems that there's not going to be that much of restrictions. Also, it seems that there's going to be several predictions per seed. And what else? What are the seeds? Well, anyway, well, let's go very quickly to some of the examples. Like, for example, this one. Let's go to this one, the protein RNA ion. In this one, you can upload the RNA sequence, and as you remember, it should be G, C, A, or U. And protein, this is all the amino acid sequence and another protein. So it seems that this complex of molecules will be based on an RNA and two proteins and two ions. So let's see, where's the example. Oh, okay, here it is. So you can see here that this is the two proteins and the RNA. And the RNA, as you can see, probably is this one that is in teal color. And actually, this is a tRNA, just based on the molecule three-dimensional structure. So that is very cool, because that way you can see actually by colors also the confidence. You can see that there's a very high confidence of this is like a kind of a score. So there are proteins that have had very high confidence score, and there are RNA. This RNA molecule, it's just confident. It has a confidence score, and there are also some regions that these the organized regions that are usually with very low confidence. But this is very cool because we will be able to generate whatever protein or RNA or DNA complex just based on inputting any molecule and any sequence that we want. So let's see another example like this one. This is it's a big protein, it seems, and it has prostalational modifications. So this modification seems to be in this part. As you can see here, this is the protein, and this small. Let's see if we can zoom in a little bit more. But in here you can see that there's the confidence probably is in blue color, and the post-translational modifications are these ones that are in teal. These are the molecules. So once again, this is very cool information. So this is like, that's why it's a breaking news, because we are just able right now to generate three-dimensional complex structure based on just inputting the sequence in just like by inputting into the server. So let's go directly with the server. I already entered to the Alphafold server. I created my job. So as you can see here, we will be able to add whatever we want, like DNA, RNA, or ligand. And we can even add more, like I want three proteins, I want to paste the FASTA sequence. So we will do later some other examples, but as you can see here, we can add the RNA sequence. So if you were to imagine, we can add A, G, C, C, A, G, 2. So I am adding here like several molecules, but you can also add whatever sequence of a protein that you want, and another protein, and even we can add like another like ion, like I don't know, like a zinc ion. Okay, so this is very cool. And then after that, I will not generate this one right now, but actually let me try very quickly by adding just a DNA sequence based on RNA. So if we were to search for one of my favorite molecules is the 5S ribosomal RNA sequence that is part of the ribosomal RNA, we can try to get the sequence. So I will just open here the 5S ribosomal RNA sequence. And let's see if I have the sequence in here. Well, I don't have it, but it could be here. This is 120 nucleotides. So if this is correctly, I will be able to get the sequence of the PDB. Let's see. So this is secondary structure, taxonomy. This is already the chain. Well, anyway, it seems that we will be able to generate any sequence that we want. But I will definitely create another video where we will be able to do more examples. But as you can see here, this is really very interesting breaking news about AlphaFold 3. So please stay on the channel. Don't forget to follow me, and just give me a like, and let's see you in another video of Moonlight Biology, Moonlight Molecular Biology. See you all next time.